let's start with the clinician. So, Dr. Siddhant, could you share an instance from your experience where there has been a clinical dilemma, and if you could throw light on how it got resolved?
So I'll start with a case that we dealt with a couple of weeks ago at a hospital I was in. So we had an extremely immunocompromised host, a CLL patient on immunotherapy, steroids, received whatnot, and coming with a lower respiratory tract infection, imaging showing very diffuse ground glassing interstitial shadows, little bit of atypical bacterial kind of a picture. Sputum didn't yield anything, the upper respiratory panels didn't yield anything. Eventually, we went in, did a bronchoscopic cultures for fungal biomarkers, everything came negative. Which is when I was called in, we were pressed for a diagnosis, patient is worsening in front of us. And at this point, when we were pushed against the wall, we asked for the sequencing time for the bronchoscopy, and I also asked for a throat swab NGS. And somehow both of these gave us the answer of HSV1 viral pneumonitis. Okay. Now, again, the next question put to us is: could this be a colonizer? Because this could be a part of the normal flora in that area. Two things which fit into this completely, that none of the bacterial panels, none of the cultures, anything gave us an answer. This was the only thing we got. Second thing is we proved that this was a viral lower respiratory infection by seeing the cytology, which had inclusion bodies, which proved that this was a diagnosis. It is not something which is available on any of the panels till date. So this is how this technology helped us get to the answer. We started the patient on antivirals, patient improved, and eventually because of other issues the patient succumbed, but at least we had an answer for what we did.
Thank you so much, Dr. Siddhant. It was insightful. So, Bansi sir, who also happens to be a senior from college, so he is an alumnus. Sir, as a clinical microbiologist, could you share your experience with respect to the technology which Dr. Siddhant just now mentioned, a next generation sequencing based assay which provided him with the answer, and the impact which you saw in your experience when you were using this for your patients at your hospital?
So thanks, basically our journey with NGS and nanopore sequencing started I think 8 to 10 months back in Max Healthcare, and we started initially with clinical validations of NGS with directly from samples and from the blood culture positive and few biopsy positive correlations, and then we have incorporated in our NABL scope in February 2025, and we have done almost 1250 samples till now in last 8 to 10 months. So I will highlight few clinical cases which we have incorporated in a fast clinical diagnostics basically for NGS. The first I will say few endocarditis patients. So basically from the so a lot of patients have blood culture negative. So we have directly from the vegetation tissue we have identified Histoplasma capsulatum, Exophalia dermatitis, fungal endocarditis, and bacterial endocarditis like Streptococcus viridans. So in routine protocol, vegetation tissue we are directly going for NGS because all the cultures are coming. Second, I will describe one anaerobic infections. We have one case of doctor's father of craniotomy 6 months back and a cranioplasty, and after small injury, then again a subdural hematoma and cellulitis, where we got NGS and acute bacteriomegalis and anaerobic infections. And we have empyema, pleural empyema where bacterial sphagnus also is. So anaerobic is another potential where NGS would be. Third case, one case interesting is Mozzarella tropical and Mozzarella mycetoma. One case, there is a 12 years, he was having sinus in the lower leg, that is from South Africa patient, and had multiple surgeries and came to Max. And we identify as Mozzarella mycetoma, and we have, I think their full calculus was missing, and the doctor told we have to do the bilotomy amplification. We have given itraconazole, but that patient lost to prolong. Another interesting surgery, basically a plastic surgery, doctor sent out was samples. Basically 1.5 months back, as a fracture femur and Then after 1.5 months, there is a slow infection coming at fracture site, and where we got Mycoplasma hominis, and we are given toxicity. Then we have another two neurosurgery cases where one patient I remember CSF sample where culture negative, BIFR negative, and we got identified Klebsiella pneumoniae, which is ESBL positive, and we given meropenem. And another case is Cinetobacter pneumoniae where BIFR was negative but culture came positive. So in these important 5 things which are during the characteristics, neurology, these deep-seated infections, and neurosurgery, patients who have the BIFR. In these cases, so we have put forward NGS as the first preferred diagnosis in our hospital. Thank you.
Thank you so much, sir. And I must also mention that we got a call from sir one night, and that was following a surgery. Sir, if you remember the fracture femur case when you placed a call, a very unusual finding from a wound infection. So we would never expect a Mycoplasma hominis in a wound infection, and I think every microbiology lab is very likely to miss this diagnosis. But sir, because he made sure that the sample reaches the sequencing assay, he could detect, deliver the result, and the rest is a better patient outcome.
Actually, every day we run around 10 o'clock in the evening, and next day 5 to 10.
Great, sir. So I think I'll move to Aruna ma'am. She again has been a very strong force in my journey as a medical microbiologist. So every time I place a call to her whenever I'm stuck at some point. So ma'am, and I think this has been because you I see as the user of nearly every microbiological technology available over the years. Where do you see sequencing in the diagnostic spectrum and why? And if you could substantiate your opinion with some clinical experience that you had.
So I think probably about 20 years ago, we never thought that we would have a multiplex PCR available to us, results available within 1 hour. If we have that now, I think the next thing that we have on our platform that should come into the clinical microbiology lab is next generation sequencing. And I had the fortune of actually collaborating on such a technique for a research project on bloodstream infections. This study we did in about 22-23, where we actually took 400 specimens from patients and who also had blood cultures. And when we evaluated it through next generation sequencing and a routine blood culture, BACTEC blood culture, we found that our BACTEC blood cultures had a positivity rate of about 11%, whereas this next generation sequencing platform gave us results to about to the tune of 19%, which I think is significant because we are struggling to get organisms or blood cultures positive, and these patients were suffering. In addition, we got antimicrobial resistant genes all upfront. So all of these results likely to come within 24 hours, whereas you struggle with your blood cultures, for 48 hours and more. So that was a very positive experience for us, and we have started using this particular technology for bloodstream infections. Another very interesting situation that we had was we had this patient who we had initially grown as Campylobacter. He was an immunocompromised individual who had grown Campylobacter in her blood culture, and both Vitek as well as Malditoff had identified it as Campylobacter coli. We had started her on appropriate therapy, and she seemed to, the moment we would stop antimicrobial, she seemed to come back again with fever, and her blood cultures on 3 consecutive occasions actually came positive with a similar, Campylobacter looking organism, but it would just not, we wouldn't be able to isolate it on solid media. And when we used this particular technology, we found that it was actually Helicobacter cinnaei and not really Campylobacter. So that ID was very, very important to us because once we started reading about Helicobacter, I had not come across it in any of the blood cultures so far in the last 20 years. So when we came to know that it was Helicobacter cinnaei, we realized that this patient was immunocompromised, so it had a high risk of such a pathogen. And Helicobacter cinnaei is a bug that requires multiple drug therapy and for a prolonged duration of time. So then, with the help of an ID physician, we were actually able to treat this patient, and she got rid of that bloodstream infection. Another thing interestingly we did was Mahua offered to do the WBS of stools because there had to be a source for this Helicobacter cinerea. And we found Helicobacter cinerea in her stool sample also. So that was another very interesting thing that we observed, something that we wouldn't have been able to, we didn't, in fact, Margitop as well as Vitek actually misidentified it as Campi coli. So this is definitely an interesting technology, and what I'm excited about is that it comes in 24 hours directly from the sample. That is what is really exciting.
Thank you so much, ma'am. I think very important message because a pathogen identification becomes the first step towards an antimicrobial stewardship. I think all of us agree, and that is the endeavor which every clinical microbiologist tries to make. It's just for us to move to a technology where this is feasible and feasible with an accuracy where a Helicobacter cinerea is not wrongly identified as a Campylobacter coli, or where Legionella longbechi is not missed because we always try to think of Legionella pneumonia. So I move to Nilma ma'am. Now, why our previous speakers represent the corporate sector and every new technology, and that has been my experience when I crossed the borders of PGI and went to work, I found how delimited most of our centers are as far as advanced technology availability is concerned. And I think we as microbiologists who have access to some advancements because of the place we are also bear an onus of taking these technologies to the masses. So I think Nilma ma'am, as a representative of service being delivered to the masses, needs to throw light on, ma'am, a diagnostic solution for the classes versus the masses. Do you see the adoption of this technology for them, and what are the steps which you would like to suggest so that this adoption and accessibility happens?
Yes, Mahua. Why not? I mean, we can actually take this to the masses because currently government sector labs, they have conventional methodologies which are there, which are foolproof. But if we need to scale up, we have to build up capacity, and we need to move forward because sequencing for the future is going to be a game changer when we look at antimicrobial resistance as well. So according to me, yes, this has to be taken forward. We could actually look at maybe just starting a little slow, starting with the critical care unit patients, and then maybe scaling up and going wider to all the other patients that we see in government sector hospitals. And finally, this could be a test which could be something which could be used as maybe even a first line, because we are looking at direct samples, we are looking at a turnaround time of 24 hours. We have clinicians today in the government sector who are asking us for reports which are more quicker, which are asking us for gene targets which are responsible for giving those resistant mutations. So I think yes, the final aim should be taking it to the masses.
Fantastic, ma'am. A very important insight on this was shared by Aruna ma'am. Ma'am, if you would just like to elaborate on that piece where you mentioned that COVID has enabled the country to almost grassroots levels in molecular prowessness, and if that can be moved forward.
Yes, certainly. I think, meaning most of the government labs as well as the private microbiology laboratories now today have a molecular facility. So the basic molecular facility is there. And a technology like nanopore probably does. You don't need to, infrastructure-wise additions are not really required. What would be required is in terms of resources, in terms of personnel who need to learn this technology and the bioinformatics, etc. But I believe the platform already has bioinformatics in it. So what's important really is, simply scaling up capacity.
Maybe one point that I want to add, Mahua, is the cost factor.
Yes.
And the moment we start scaling up and using this kind of a technology on a wider platform, the technology industry itself might have to put down their costs, or it would facilitate putting down their costs.
I'm sure, ma'am. I think that is the advantage we have when we are indigenous. That is the scope an indigenous company always has, better than anything that is brought from the Western world. So this is for the students over here. When I was a student and I used to hear about advanced technology, it seemed to be a distant dream. But trust me, with what ma'am has said, nothing is a distant dream, so please don't stop dreaming. I think now I come to somebody who bears the biggest onus. So he is the scientist and he is the technology enabler as well. So Anirban, you have heard the real world scenario from the experts representing the disparate healthcare ecosystem of the country. So could you throw the light on the following aspects, and I think I put forward the most difficult questions to him. What is the potential of this technology that you see, the current limitations that you perceive, and what are your suggestions on the ways to overcome and increase adoption?
Thanks, Mama. Basically the question that you have asked seems like I need to foretell the future. No matter what I tell about the future, you will interpret it the way you want to. Really doesn't matter. But what I can definitely tell is where we are today and where we started from. So Haystack as a company started from IIT Bombay not so long ago. So there's an incubator in IIT Bombay, literally like a fetus. They incubate us. And when we started, and I am so thankful to people who are here and others who are not here right now. Just to give you an idea, our first 100 samples was for tuberculosis, and all the 100 samples were used for validation of the technology at the JJ hospital under the guidance of Dr. Anita Joshi and Dr. Nilma. That study was published. At that point of time, the sequencing of one TB genome used to cost us 15,000 rupees, and it used to give resistance profiles of 13 antibiotics, and that's about it. Today, our TB test can give resistance profiles of 18 antibiotics. The cost of the test is 50% lesser than where we started from, and we can detect non-tuberculosis, mycobacteria, and mixed infections. The first sepsis sample that we picked from was of a patient in Breach Candy from Dr. Aruna Pujari's lab. At that point of time, that sample cost us 1 lakh to test. It took us 72 hours, and we were doing only blood. Today, the test is available for 1/10 that cost. The turnaround time is actually 24 hours and coming down every month that we do. And today, at that time, it was just one fungus. We can now actually screen for all bacteria, all fungi, antimicrobial resistance, several of the viruses in that single test at 1/10 the cost what we were doing then. The first time we dared to take this test beyond our lab, that is taking sequencing to a lab which has not done sequencing ever. And we think that this was the first instance of distributed sequencing, which is unsupervised sequencing by the scientists that we were, was with Dr. Ansita Tarai's lab at Max Hospital. And at that point of time, when we took it to Dr. Tarai's hospital, we were still doing, I think, 3 sample types. And today, that was then it was one distributed lab. We have now gone through more than 15 of them. And we're now doing more than 32 samples. What I want to say is, if we could come down from 1 lakh per sample to 1/10 the price, if we could come down from 72 hours to less than 24 hours. As a company, which is right here, starting from IIT Bombay, the need of the hour is to get 2 hours. I've been told I need to give a result in an hour. We'll work towards it. Let's see where we get. And one thing that separates us from a lot of people who are bringing technologies from outside is we're listening to all of you. My technology, Dr. Sanjay Bhattacharya was here. He held us in all the fungal detection. So we are here. Our next targets are to bring down the turnaround time even more. We are increasing the viral panels, and I think we are challenging the turnaround time to a limit where we are now working with the government of India and BIDAC to actually get this technology manufactured in India's largest medical devices space. And of course, we are integrating AI. I can't promise you when all of this will be available, but to your question, Nirma, I think, Dr. Nirma, I think we are working towards a lesser cost. To your question, Dr. Aruna Pujari, we are working towards more rapid tests. And Dr. Siddhant Shetty, who is now set us on the way to understand the clinical side and what the patients need more, will be working towards that. And I'm sure Dr. Bansidhar Tarai will be guiding us more on some of the automations that we have already worked on with you.
Thank you so much, Anirban.
I just wanted to make one comment that 1 lakh rupees was not paid by the patient, it was borne by his side.
Thank you so much. That is where we probably as a company invest a lot in bringing about good patient care. I think we're very hard pressed on time today. So for any sort of questions, doubt clearance or further discussions you want to have, you can take it over tea or over the banquet. Thank you so much, speakers. But what my takeaway from what I hear today and what I have learned as a student is this test is a go-to test. This is not the future, it is the present. So if we want to break barriers, if we want to modernize medical microbiology, this is where we should be. Thank you so much. Thank you so much.
Thank you.